I want to welcome everybody to the International Elephant Foundation's Conservation Chat. We're really glad that you've joined us today, and I know that there's people from all corners of the globe for what promises to be a fascinating discussion about elephant conservation. For those of you who don't know me, my name is Julie Bates, and I'm the Donor Relations Manager for IEF. And other staff members we have on the line with us today include our Executive Director, Deborah Olson, Sarah Conley, our Director of Conservation, and Dr. Chase Ledoux, our Special Projects Manager. So we all welcome you today. A few housekeeping notes before we get started. Let's go over a few quick things. Make sure that you have your microphones off. It will help minimize background noise and make sure that everybody can hear the speaker. Cameras, please turn those off as well once we get started with the presentation. That will help us save bandwidth because I know some people are tuning in from other parts of the world where they might need some extra bandwidth. Also, just a note, we will be recording this chat. And also, if you have questions for our presenter, please put those in the chat box and we'll get through those after about maybe 30, 45 minutes. And I think that covers it. So without further ado, I'm going to turn it over to Sarah, and she's going to introduce our presenters.
Thanks, Julie. Hi, everyone. Thank you for joining us. Today, we are joined by two incredible researchers, Doctors Joshua Schiffman and Lisa Abegglen. Dr. Schiffman is a pediatric oncologist and a cancer researcher at the University of Utah and Huntsman Cancer Institute. He co-founded PEEL Therapeutics, a company that develops new therapies based on evolutionary biology. Dr. Abegglen is a scientist at the University of Utah and Huntsman Cancer Institute, and with expertise in comparative biology. Together, they lead a translational research lab focused on cancer susceptibility and resistance across species. They're internationally recognized for discovering that elephants have extra copies of the TP53 gene, which I'm sure they will tell you more about, which may help protect them from cancer. Their current research investigates immune responses, particularly things called NETs, which we'll hear more about, and so I won't try to tell you. And so there's so much that elephant conservation and human medicine interact and hopefully can benefit each other. Today, they're going to share their latest research with us. So please welcome our friends and innovators, Josh and Lisa. Hi, guys. Thank you for joining us.
Thanks. Can everybody hear us okay?
I think we're all set.
Yep. Okay, great. Well, it is a real treat for Lisa and I to be here with you this morning. One of our favorite things to have conversations and chat about are elephants. So what better way to start off the morning here in Salt Lake City, Utah, than having a chat with all of you around the world about elephants. And let me just take a moment before we dive in to explain how Lisa and I came to be so interested in elephants. What's our elephant origin story? And so you heard, thank you for the beautiful introduction. You heard that we are researchers. I'm actually a pediatric oncologist by training, and so obviously quite interested, curious, and concerned. Where does cancer come from? Why do people get cancer? And over a decade now, makes me feel very oldly. So over a decade now, speaking with colleagues, we learned what many of you have already observed in your own careers, that elephants almost never get cancer. Certainly they do, but at a much, much lower rate than we would expect, and certainly a much, much lower rate than humans or many other animals. And that led us to try to understand why and to meet all of the people in the elephant community. And so, as you can see, these elephants, they have an enhanced cancer defense. And why is that? What's the evolutionary explanation for it? Well, if you think about cells in the body, every time a cell divides is an opportunity for that cell to develop a mutation and turn into cancer. Now, elephants have 100 times as many cells, so that's 100 times as many opportunities for those elephant cells to turn into cancer. Now, you can look at it a different way. Elephants, as you know, take 22 months of gestation, and those baby elephants are born anywhere from 200 to 300 pounds and sometimes reach as much as 10,000 pounds in less than 10 years before they reach reproductive age. Again, this is like bringing coal to Newcastle, right? You guys know all of this very, very well. But if you imagine a small, well, relatively small, 300-pound elephant growing to 10,000 pounds in 10 years, that's putting on almost 3 pounds a day, 3 pounds of cellular mass growing. So much cell division, you would think that all of these baby elephants would be dropping dead from cancer before they could reach the age of reproduction. And of course, if that happened, elephants would go extinct and there'd be no more elephants. So elephants throughout evolution, we believe, had to evolve some way of avoiding cancer. Hopefully that makes sense to many of you, and you've probably heard Lisa and I talk about this before. And in fact, our first real encounter with the international elephant community in IEF was, again, over, wow, 2013. That was a different world we were living in back then, at least. But anyways, where we began to present and share our work. And Lisa led a lot of efforts. I'll turn it over to her in a moment. She can comment on this slide if she has anything to add. But led us to really working with colleagues who also helped to make this discovery that elephants have extra copies of P53, which is a cancer-fighting gene. So all of us, hopefully on this call, has 2 copies. Those 2 copies are enough to pretty much keep us safe from cancer for a little while. As we get older, of course, the risk for cancer goes up. But with the 20 copies, or rather 40 copies, 20 versions of the elephant P53, they're able, we think, that contributes to their ability to decrease cancer. And as Lisa has been working, and I and others, we found out that the elephant P53, even 1 copy of elephant P53 versus 1 copy of human P53, seems to be much, much better at getting rid of cancer cells. So this is very exciting work. So Lisa, I don't know if you have anything else to add to this.
No, I was just going to say that it was at these meetings that the International Elephant Foundation hosts where we learned about the project we're going to talk about today, which is EEHV. I'm going to let Josh continue.
Yes.
Introduce that.
And I will say also, just before we talk about EEHV, which you guys know about, it has been just to my radical amazement and joy to be able to work with the elephant community, right? So here I am, a pediatric oncologist taking care of kids with cancer, running a research lab. Lisa also helping with this research lab, pretty much running it as well. And we had never dreamed, you know, when we started our careers that we would be so involved with people taking care of elephants, and that elephants could teach us so much about human disease, and human disease, as you're going to hear in a moment, may actually help us with elephants, and importantly with conservation. So it was at these meetings that I learned actually from, I think it was Dr. Howard, our friend Dr. Howard, who was presenting about EEHV. And of course, you know about this very well. This is a devastating threat to elephants under human care, but also in the wild. It's a viral pathogen. It creates vascular injury. When they have this infection, it activates the platelets. It leads to this phenomenon, this bleeding diathesis called DIC, right? Disseminated intravascular coagulation, where you get hemorrhage and shock. So these elephants are clotting and bleeding and clotting and bleeding, and often they can die within 36 hours of developing signs. It happens most often in younger elephants, and it is one of the major causes of mortality in Asian elephants. And so it is a conservation concern, right? Because I just told you, 22 months, right, to bring a new elephant into the world. And then, you know, within hours, that can all be just gone. And of course, we all know elephants can live 50, 60, 70, some even 80 years. And to have an elephant's life cut short is tragic, especially not just a majestic animal, but an endangered animal. Now, when I first heard the description and actually saw some of the pathology slides, again, I think it was Dr. Howard who was presenting, I said, wow, this looks remarkably like disease that impacts the patients that I've taken care of, right? Especially immunosuppressed children. And so I began to think, is there a connection here? Is there a connection here between what's happening, what I've seen in humans patients with this DIC, and then with what this viral infection that these elephants get with EEHV. Okay. So now we're going to talk a little bit about neutrophil extracellular traps because that we think may be bridging the connection between the two. So it turns out when neutrophils, which are part of our immune system, get exposed to pathogens, they're almost like Spider-Man cells. When I went to med school many, many years ago now, we were taught that neutrophils were like Pac-Man. They gobble things up. And Lisa is much smarter than me, and she's got a PhD in immunology. And again, this is what they taught, right? This was the canon that neutrophils, Pac-Man, gobble things up. But it turns out they actually also shoot out sticky webs. Like I said, almost like spider-man. And those sticky webs are the DNA inside these immune cells. And those sticky webs actually catch the bacteria and viruses and pathogens that are floating into in the bloodstream. Now, a little bit of these nets are good because they clear out what shouldn't be there. But too many nets cause inflammation and blood clots. And in fact, many diseases can be attributed to these nets. There are acute diseases, sepsis and DIC, thrombosis, stroke, myocardial infarction, even acute respiratory distress syndrome, which we'll talk more about. So this acute inflammation leads to really this cascade of clotting and bleeding and clotting and bleeding. And there are also some suggestions that chronic diseases, Alzheimer's disease, obesity, multiple sclerosis, atherosclerosis, all of these nets from these immune cells may contribute. Now, what was really striking and almost the aha moment was when COVID hit, right? So we were doing our elephant research with EP53 and trying to understand how EP53 is preventing cancer in elephants. But we began also on the side to start thinking about neutrophil extracellular traps in the immune system and how does those nets contribute to inflammation, maybe even cancer and metastasis. And then after COVID, all of these patients began to develop this DIC, right? This acute respiratory distress syndrome. And you'll remember in the beginning of COVID, I don't want to be too controversial, but I'll say before the vaccines, when many patients were hospitalized and died, a lot of the deaths were due to this respiratory distress. And this respiratory distress, we recognize in our group at the University of Utah with Dr. Yost and others, Dr. Middleton, realized these patients with these blood clots, these microthrombi, these mini blood clots in their lungs, it was very similar to what we were seeing with other net-related diseases, and we were able to look at autopsy specimens from patients who unfortunately were dying, again, pre-vaccine, were dying of COVID, and we found, sure enough, many nets in their microthrombi, in their mini blood clots, in their emboli, in their lungs. And the more nets that they had, the worse the COVID-19 symptoms and the worse they did. And of course, we were continuing this time our connection with many of you on the call, right, with IEF. And this is just this is a love story, right, between humans and elephant researchers and caretakers. And we began to say, you know, this EEHV, when the elephants die, it's very similar to these patients who are dying with this hypercoagulability. And we began to wonder, could there be a connection between nets and EEHV hemorrhagic disease? Do you want to take?
Yeah. So,
all right.
So there is some evidence that there's microthrombi in EEHV. These are a couple of papers that were published fairly recently showing evidence of microthrombi in tissues from elephants that succumb to EEHV-HD. And there's also evidence that DIC is occurring. And DIC, I think Josh has already explained pretty well, but what we know that nets are involved in DIC, so you can get activation of the neutrophils through viruses and other types of activators, and it releases these nets. Then it actually captures red blood cells and fibrin and other parts of the cascade that cause these microthrombi. So we wanted to determine if this could be playing a role, this net release in EEHV. So we needed first to figure out how we were going to test if elephant heterophils actually could release neutrophil extracellular traps. So heterophils are the equivalent functionally of neutrophils. They're just called heterophils in elephants. So we needed to collect blood from elephants to do these studies. The heterophils are circulating in the blood. And fortunately, through our work on cancer, we had established a very incredible collaboration with our local zoo. This is Utah's Hogle Zoo, and it's just like less than a mile from our lab, which was really important. So we were able to collect that blood and begin doing some studies to determine if the heterophils from elephants could indeed release neutrophil extracellular traps. So it was very, very critical that this blood that we collected be fresh. Neutrophils are very short-lived, and if we didn't process the blood within 30 minutes of the draw, the experiment would fail because the neutrophils would have already released their nets. So we needed to keep that from happening before we initiated our experiment. And this just tells you how we did that. We did red blood cell sedimentation, and then we put the cells over a gradient and we were able to isolate the heterophils, and then we could put them into our assays. So we did cytospin to check their purity, and then we're able to measure net release from these cells by stimulating them. And then there are antibodies that we can add that actually label those nets. And in the experiments I'm going to show, we're labeling those with an antibody to something called citrullinated histone H3. So when nets begin to be formed, the DNA decondenses and the histones get citrullinated. So it's a really nice marker of net release.
And these, by the way, these are healthy elephant heterophils,
yeah.
right? So we're starting with the healthy ones just to show that they could actually make nets. And then we'll go on and we'll talk about EEHV.
Yes, that's right. So in human research, we know that there's a number of different stimuli that you can treat neutrophils with, and that will cause them to release these nets. So we wanted to test that first. We isolated the neutrophils and we added, in this case, ionomycin. And what you will see here in this video is that elephant heterophils do indeed release nets. You can see this beautiful webby-like strand of DNA being released from this cell, which was very exciting to us. And it's the first demonstration that elephant heterophils can release nets.
Right. And again, this is all happening in a dish.
You're right. That's right. So next, we wanted to test some other stimuli. So this is PMA, LPS, which is a part of a bacteria that can stimulate these nets, and ionomycin. And we tested, these are African elephant heterophils. Those are the Utah's Hogle Zoo had African elephants. So we tested it and we looked to see if there was a dose response, and we did see a dose-dependent response, which was really exciting and suggests that these cells are behaving similarly to cells that have been tested previously in humans. And then next, we're very interested in testing Asian elephant cells because Asian elephants seem to be somewhat more at risk of developing EEHV compared to African elephants. However, there are not any African elephants in Utah, and so we had to figure out how we could get that fresh blood. And fortunately, through actually attending the IEF meetings and establishing collaborations, we had a very strong collaboration with Houston Zoo and Paul Ling, and we were able to reach out to them to find out if we could actually travel to Houston to perform some experiments. And it was really important also that Paul Ling's lab at Baylor is very close to Houston Zoo. So we were able to have that blood collected, transport it to Paul's lab very quickly, and then we were able to do those experiments right there in Houston, which was really, really incredible. And we were fortunate that we had this collaboration established. So we did the same experiments on the blood from the Asian elephants, and we found, again, that when you stimulate them with these known stimuli that are known to cause neutrophil extracellular trap release, that the Asian elephants are also releasing nets.
I just want to, Lisa's used to me interrupting.
Yeah.
So yeah. I just want to pause for a minute, just for everyone on the call, because this is more of a casual conversation. I mean, it's a scientific presentation, but we're sitting at a table. It's not a lecture. Just pause for a moment and just think about, again, I just come back to this concept of radical amazement of this idea. Here we are, two scientists. We're trying to study elephant cells. We only have African elephants in Utah. They're Asian elephants in Houston. These elephant cells, they only live a few hours, so you have to get them right away. And because of these collaborations, because we're friends, we're colleagues, we're scientific peers, we were able to, and again, I credit Lisa with all of this relationship building with all of you to really establish that trust, right? To come and do this work together. A group of scientists get on a plane from Utah. It sounds like a joke, right? To travel to the Houston Zoo to grab elephant blood, and then to work on it in a nearby lab. So really, it is the scientific community. It's everybody working together that really, I just want to emphasize, makes this amazing science even possible. Okay, now back to the science, Lisa.
It's a really good point, Josh. And there were more people involved. We recognized that we weren't the experts in EEHV, so we assembled an EEHV net working group, and many people really contributed and guided us as we were doing this work, and we were very thankful for that. So next, we wanted to know if EEHV HD plasma could stimulate net release. So we know that with humans, when there's an infection, that the plasma from the blood of patients that are affected can stimulate net release. And here you can see I've already given away the answer. When we stimulated these cells with the plasma from EEHV affected blood, we did see net release, suggesting that there's something present in the plasma that is stimulating this response, and it could be contributing to this worse disease state. So the next thing we wanted to know is there evidence of nets in the tissues of elephants that succumb to EEHV-HD? So we were able, again, to collaborate with a number of different zoos and collect those tissues and do the testing to see if there was nets there. So this is what tissues should look like in elephants that are not, that when there's very little nets present. So what you see in this picture, the blue, which has been the same in all the images, is going to be the normal DNA from cells. So it's usually just labeling the nucleus. The nets are in the green. That's the citrullinated histone H3 that I mentioned earlier. And we've labeled neutrophils with an enzyme called MPO, myeloperoxidase. So in normal tissues, you shouldn't see very many heterophils and you shouldn't see very much evidence of nets. And that's what we see here. So these were tissues from elephants that died from other causes. But when we looked at the tissues from the elephants that succumbed to EEHV-HD, we saw a tremendous increase in both heterophils and nets. And we even see evidence of what looks like some microthrombi here in the heart. So we collected this data and we looked at tissues from a number of different elephants and we quantified it, and we found that there is indeed a significant increase in nets in the tissues from elephants affected by EEHV-HD.
And I'll just also add, clinically, as many of you know, these are the same organs that are so impacted with elephants with EEHV hemorrhagic disease, right? Strokes in the brain, respiratory failure in the lungs, liver failure, cardiac failure. So the same organs that get impacted in infections of EEHV-HD seem to have the same organs seem to have this increase in these nets, in these extracellular traps.
Okay, so next we wanted to know if we could inhibit this response. So there are some drugs that are able to specifically inhibit nets by inhibiting an enzyme called PAD4. And that drug is BMS P5. And there's also a way to inhibit nets using a general immune suppressor called dexamethasone. So we wanted to know if we could inhibit this response by treating with these drugs. So that's what we did here, using those known stimuli that I mentioned earlier, PMA, ionomycin, and LPS. When we treated with either the more net-specific inhibitory drug or the general immune stimulant, we saw that we were able to block this net release, which was incredibly exciting when we saw this. And we also wanted to know if we could inhibit cells that were, could we inhibit this response when the cells were stimulated with the EEHV plasma? And again, we saw similar results where if we treat with these drugs, we're getting this really nice inhibition, which suggests that blocking net release could be a beneficial treatment in the context of this disease. And this is just the combination of that data. And I wanted to share that we have recently posted this, all of the data that I've shown today to bioRxiv if you'd like to take a look at that. We've also submitted it for publication and waiting to hear back. So what's really, really exciting about these results is that in animal models, so these are mouse models, there's really tremendous evidence that blocking nets has a huge survival benefit in animal models. And I want to show you 3 examples. So this first one is looking at hemorrhagic shock and sepsis in a mouse model where the mice are not able to make nets because they actually do not have that enzyme I mentioned, PAD4, at all. So when the mice cannot make nets and you introduce this disease, hemorrhagic shock and sepsis, most of the mice, or in this case, it looks like about 50% of the mice will not survive. But if the mice cannot make nets, then nearly all of the mice survive. So that really tells you that this nets are contributing very negatively to the outcome in this disease. And in another example, in this case, the mice were treated with a drug that inhibits nets. And this is a microbial sepsis model. When they aren't treated, all the mice die. But if they are treated to block net release using this specific inhibitor, the survival increases to 73%. So that's huge. And again, telling us that these nets are really a huge contributor to death in these models. And one more last model that I wanted to share. This is a sepsis model where they treated with DNase. So DNase doesn't inhibit net release, but it's able to chew up those nets after they're released. And in this case, they actually administered the DNase rectally, and they found this huge survival benefit even in this case where they're using rectally administered DNase. So this is really exciting, and it suggests that there's maybe an opportunity to improve survival also in elephants that are affected by EEHV HD. So just to quickly summarize, what I've shown you is that elephant heterophils release nets. EEHV HD plasma triggers net release. The tissues that we looked at that were from elephants that succumbed to EEHV had excess nets and evidence of microthrombi. Nets are likely contributing to the DIC that other researchers have seen in the context of this disease. And we found that net formation is blocked by both dexamethasone and BMS5. So some future considerations in humans, there are net assays are being considered as biomarkers of disease severity. And I know that that's something that would be really beneficial in EEHV to look at in the future. And then I wanted to mention that that's something that would have to be considered for dexamethasone. I know that that's been used in the context of EEHV before, and the results are mixed. And we don't really know if it would be beneficial in EEHV because in some viral diseases like CMV, dexamethasone can actually increase viral replication, and you wouldn't want that. And also the fact that it's decreasing the entire immune response might also be a problem. So it's hard to say if it could be beneficial or not. And then until we can culture the virus in vitro, it's hard to really evaluate that outside of a whole animal model, which is challenging. However, there are many drug companies that are working to develop net-specific inhibitors, and I think that could be a really great option in the future when those are available to reduce potentially the clotting and the organ damage. And then, you know, overall, I think that our data suggests that in the future, this could be something that could be considered as a therapeutic strategy in EEHV. Anything else, Josh?
No, just to thank Lisa for leading this effort and all of the hard work and science she's put into it. And then, of course, you see all of the zoos, many of which you guys are affiliated with, the EEHV networking group. Of course, Dennis Schmidt and Wendy Kiso have been our fearless team members along the way. And really just thank the community, because together we're making a difference for elephants and maybe even for people too. And what a great way to, like I said, begin the morning with this conversation. So we'd love to open it up to questions, feedback, conversation, ideas, and thank you.
Yes, thank you.
Thank you, guys. That was so interesting and wonderful. And we're already getting people in the chat who are saying how well organized and well communicated these ideas are. And I know for for lay people, sometimes these things can be very technical, but you communicate it so well. So why don't we start by talking more about your partnership with Hogle Zoo and Houston Zoo and all of that sort of collaboration. And Josh, you touched on this, why it's so important to have that we're all in it together mentality versus, that kind of tribalism that people sometimes do.
Yeah, I mean, I will share from my perspective that it's really important to gain trust, right? And for people to understand and for the vets at the Hogle Zoo who we've worked closely with to understand we're not there to, it's not ego driven, or we're not trying to make a name or get something off the backs of other people. It is true collaboration, right? We are just as interested in elephant health, in benefiting elephants. And it's never, okay, we're going to come in and just hand off the sample of blood that you've drawn and we'll never see you again. We work really hard. I don't know how well we do, but we work really hard to try to keep our zoo colleagues updated and part of the conversation and co-authors on the manuscripts, right? This is absolutely a group effort together, and again, with the common goal of benefiting elephants.
That's awesome.
Yeah,
And,
go ahead, please.
Oh, and being able to access elephants in human care is enabling this, right? Having
Yes.
Yeah.
Yes. Yes. And I don't know if everyone knows, but our elephants in Utah no longer live in Utah. Now they live in Kansas City. So yeah, it's challenging for us to continue our studies without traveling to where the elephants are.
Yeah, and it's true. At the end of when we were wrapping up these studies, there absolutely was a clock.
Yeah.
There was a countdown, and one of the things also important, and I think people on this call understand, but we always have to explain, especially with our original work, there's a lot of misconception in the public, which made it very challenging initially to talk about our work, because people would say, "Oh, you're torturing these elephants. You're putting cancer cells in their bodies. That's how you know they don't get cancer." And
or you want to use their blood for treatments, which is
that's right.
Never been the case.
That's right. The other, we didn't talk about, maybe we'll come back on another conversation, talk more about the P53 work and the medicines we're working on there. But we studied the elephant blood, and Lisa and others in the lab, so that we can start to make synthetic elephant P53. Our goal is never to give people elephant blood or bleed elephants dry and treat people that way. And the other thing that really took some education was we really worked very, very hard to try to time our experiments around blood draws that are already happening in the elephants, right? So we're not trying to poke needlessly or extra times when we can avoid it for our research. We piggyback on, oh, this elephant is getting a blood test. And of course, many elephants do get in captivity, right, in the zoos, are with human stewardship, do get frequent blood tests for their own health, for other purposes. And so then we work together at the same time to say, okay, well, let's coordinate.
Yeah.
That's great. And it's all about increasing welfare across the board, which is so important. So we have some questions in the chat. What is the trigger to net release in vivo?
And for EEHV, I assume that question?
I think so. The follow-up is, may cytokines be involved in that? Perhaps?
Yeah, it's a really good question. The answer is that we don't know exactly what it is, but there's a number of different factors that can trigger net release. Cytokines certainly one, virus can do it, and platelets and the bleeding cascade.
The endothelial cells, the lining of the blood vessels, right? Those are very sensitive to any type of damage response, and there are cytokines related to that damage. And when the endothelial cell gets damaged or nick, then it releases basically signals that then trigger the nets, the neutrophil extracellular traps, or in the case of the elephants, right, these heterophils extracellular traps, to be released. So it's really a confluence of damage and pathogens.
Okay. And is that general across the board, say outside of EEHV? Like, why would these be released, or in other species? Are there different mechanisms for causing them to be released?
Yeah, before we started this work, and I actually was hoping to do a cross the species study for neutrophil extracellular trap release. Turns out that measuring neutrophil extracellular traps is really, really challenging. You kind of need a lot of blood, so it's harder to do the studies in smaller animals, and there's this timing issue where you have to test basically the blood immediately. But we didn't know, and we have this great study now with the elephants, and so far for the elephants, the same things that trigger it for humans is triggering net release in the elephants too. We haven't tested every factor, but those general factors that are known are stimulating net release in the elephants also.
And there's also this concept Lisa's also alluding to. In evolution, you may evolve one mechanism to benefit something, but then it actually has a detrimental effect, right? So again, you on this call are the elephants experts of the world, right? It's in your name, the International Elephant Foundation. And many people believe, people I've spoken to who work with elephants have said, elephants in general tend to not get as sick as often. When they get sick, they get very sick, right? When they have this EEHV, but for the most part, they're not coming in and out of the vet's office at the zoos so frequently, and they seem to be generally healthy, not immune to infections, but they seem to be able to handle it. Now, that may be because they evolved this immune system where they're a little bit more likely to trigger this net release, right? So in the normal functioning immune system, that's good because that may help actually prevent them from getting excessive infections and lead to their longevity, right? It may even an increased immune system, not inflammation, but an increased immune system, which then would decrease inflammation, may also contribute to lower rates of cancer. But because they're set up to have these increased inflammatory responses, to prevent long-term inflammation, that may be the triggering event for EEHV hemorrhagic disease.
Interesting. Okay. More questions. So say you have a sick calf. Is there some sort of test that you can do to see the net levels in their blood, or is that not something that's easily done?
That's a great question, and something that we were really interested in developing. So there isn't one because we worked on optimizing the right antibodies for that kind of an assay, and it can be developed certainly with more work. Yeah, we basically need the antibodies to be able to capture those nets in addition, then measure and quantify them accurately. So it would just take some more work to really optimize that assay, basically.
We've seen in human medicine, right? There hasn't been yet a standardized net test. Although we hope that people are working on one, on some, and they're being developed. One of the challenges is trying to test it right away. The other thing you can test, and we actually published a paper on this related to COVID, is that and this came through PEEL Therapeutics, that instead of measuring just the nets themselves, the net levels, because those can be artificially high or low as the body breaks them down, you can measure the responsiveness of the actual immune cells. So how likely, what's the capacity, the ability of the individual immune cells to be spitting out nets? And if you have, say, a sick calf who's got a EEHV, we would predict their immune cells are going to be firing many, many nets, and you can measure that ability. But these tests are still all research, right?
Yeah.
What we would need to actually do this is massive collaboration and able to collect the blood and store it the right way and then test it as we move forward.
Okay. Yeah. Well, more avenues for research. So just back to some foundational stuff. Is there any physiological benefit to nets? Like, why did the body create them to begin with?
You're the immunologist himself.
So they're your first line of defense against pathogens. So when the cells encounter a bacteria or virus, they shoot these nets out and they capture them, and then they can digest them, and so that it decreases the issues that you have with an infection early on until your adaptive immune response, your T cells and B cells can kick in and really take care of the that infection. So it's your first line of defense. It's actually very important, and it's a bit unfortunate that it becomes so dysregulated that it causes these big problems, and contributes to clots and these other types of diseases.
Okay, so at some level they stop helping and start
Yeah,
causing trouble.
they can become overactivated, whether there's too much bacteria for them. I don't know.
Yeah, I always say a little bit of nets is good.
Yeah.
Too many nets is bad.
Right. That's a good way to put it.
That makes perfect sense. Circling back to P53, someone's asking, if you were to synthesize a synthetic P53, how do you do that? What's the process?
Oh, that's a very good question. It's basically basically it's putting the bases that build the DNA in the right order and having them fused together in the right order. It's molecular biology that scientists have been doing for decades now. It's very, very common to build a gene in a dish and then test it in cells.
You build what's called the transcript,
Yeah.
right? And then you can, once that transcript gets into the cell machinery takes over.
Yeah.
You're just like Lisa, you're basically putting in the DNA code. So you map out the DNA code and then you put that into the cell, whether it's a cancer cell in the dish or a normal cell, or maybe one day even in a patient, into a tumor cell. And then the cell itself builds the actual protein. You go from
Yep.
DNA to RNA to protein, and then you have your elephant protein, which is identical to the protein that the elephants made, but you don't need to start with the elephant blood,
right,
to do it.
Once you have the sequence, you can make it yourself.
Wow, that would be amazing. So at the end of your slides, you did talk a little bit about some potential treatments and protocol. Have you implemented any of these outside of like mouse models in actual elephants, or no, theoretical for right now?
Yeah, where my medical degree is in people, not elephants. MD, not an E.
And I'm just a scientist.
Elephant degree. So no, but we're obviously very happy to work with vets or elephant keepers and share our perspective on that to make sure that it works.
Yeah, absolutely. Because I know people, when you have a sick elephant with EHV, they're throwing everything at it that they can, trying to help this animal. So I'm sure that this will be something that people will be interested in pursuing.
Yeah, I think it would be really interesting to gather all the data related to dexamethasone, because it's definitely mixed. Sometimes it seems beneficial and sometimes it's not. And that, they kind of see that in the human studies too, but the timing of when you administer dexamethasone is absolutely critical. So like collating all of that data to better understand what's going on with dexamethasone might be helpful. But as I mentioned, dexamethasone may not be beneficial if it turns out that it's increasing the viral replication, which could be studied if you're administering that by, collecting before and after samples. Although you may not know if it's just that the virus is replicating more because that's the stage it's at, or if it's related to the dexamethasone. So it still could be challenging.
Yeah. Okay. Well, that's really interesting. Hopefully we can get more answers. So how far away do you think we are to using that protocol if we gathered some collaborators?
I think I don't know. We need a vet maybe to answer that.
Yeah, I mean, it could be immediate. Look, in human medicine, we like to do randomized controlled trials and to study the data closely, and we could certainly do the same type of thing in elephants, right? Set up a trial. We'd have to have willing participants. Again, we hope obviously with the vaccines that are coming on board that this is going to be less frequent, but when it happens, just like you said, you're throwing everything at it. And so if elephant keepers and zoo vets and zoos themselves are willing to take the risk, we could see, right? We'd have to design the study the right way. But if we're using medicines that have already been approved and are safe, for the most part, right? We know like dexamethasone, we can give this to elephants, then we can do a trial and we can see and we can collect the data. We may not save every elephant, but what we learn hopefully could then lead to something that could be therapeutic in the future.
And that other drug I mentioned, DNase, that is approved also, Pulmozyme. I don't know at what dose and how expensive that would be to try in an elephant with rectal administration, but
yeah, and Pulmozyme is a great idea, right? That seems to have less systemic side effects than dexamethasone. Um, I feel like dexamethasone is going to be a drug that's going to be helpful, but it's going to be the timing. You can do a combination of dexamethasone plus this DNase. Yeah, so could work. Yeah.
Awesome. Well, we actually have Dr. Rutan in the chat who is saying that it has been occasionally shown to work in EHV HD, specifically in Thailand. So definitely some potential. And Lauren is here also, Dr. Howard. Pilot studies in healthy elephants may be the next step. We'll see, either rectal or dexamethasone. So we'll see how that goes. Let's segue into, what do you think elephant care institutions, zoos, other elephant holding institutions can do to help this research?
Well, for us, it's access to the tissues, right? So, like I said, Lisa has done an incredible job of connecting with each of you and sharing our research. And so when there are samples that become available, and elephants, unfortunately, if there's a necropsy, if an elephant has passed away, being able to access those tissues, to access them as quick as possible. We've actually been able to generate living cells that we can then test in the lab, right? So what we're kind of dancing around here is, how do the elephant cells respond to the virus? And so that's hard to do in an elephant, right? Your elephant is, we don't think of the elephants as a very large petri dish. That would be difficult. But we could actually have a real petri dish in elephant cells, elephant immune cells. We're getting closer. Other people have worked on induced pluripotent stem cells and actually working with the living tissue then to try out these different types of responses. So the answer to the question is to keep the lines of communication open. And if there is an elephant procedure, or if there's a necropsy, to get in touch with us. Also, PEEL Therapeutics, it has a biobank that we're building out. So just elephants are so majestic and so incredibly rare, and the opportunity to study the living tissue and the biology is what's going to make an impact and allow us to move research forward, again, to benefit both humans and, most importantly, elephants.
Great. Yeah, I think the more people know that they can help, especially in those opportunistic ways, the better. So another person is asking, has any of this been implemented in humans? Maybe they're talking about the P53, but in general, do you want to talk about any of that type of interaction?
Yeah, well, there's a lot of studies. Lisa showed some studies in mice, but there have been studies with DNase. We know dexamethasone is used quite frequently in the acute respiratory distress syndrome related to COVID. And so again, it's really this COVID has shined a light on this pathology of neutrophil extracellular traps. And people are thinking of different ways of blocking that. There are studies that are still more preclinical. They're really on the cusp of clinic, of getting into human patients, to actually targeting blocking nets. what we have today are kind of general approaches like the dexamethasone to tamp down the immune system, which then subsequently leads to lower nets. So that's for the net effort. Then in terms of the P53, that work is still ongoing, making medicine from that, and hasn't yet reached to patients in clinics.
Okay, well, that's good to know. I think so. Dr. Rutan, if you would like to unmute yourself, I think if you have a question, you're welcome to answer it. I'm not sure I am able to synthesize it properly.
I'm not sure. I did ask a question, but well, in fact, I put a claim there, or a claim, not a claim. It's just a fact. There's no virus has never been isolated. So whatever you and not cultured either. So whatever you want to do, it's hardly possible in vitro.
Yeah,
yep, I totally agree. It's a huge drawback.
Yeah, it's a big challenge, which is why the plasma work, right? So to use plasma that presumably has the components of the virus still in it, or the inflammatory response that the virus has triggered to trigger more nets is so critical, right? And again, getting back to the comment, well, what can people do to help, right? Obviously, we can't generate a sample in the lab that has a virus in it. We have to go directly to the source.
Very true. And that is one of the challenges with EEHV is that we can't isolate and culture it in the lab.
Yeah, if we could do that, then it would, we could test at least what I was saying before, the effects of dexamethasone in a dish. Like, does it increase viral replication or not? But it can't be cultured yet. So that's not an experiment that we can do. That's unfortunate.
It really is. But like you said, we have the vaccines that hopefully will reduce the instances of EHV-HD. And then the ones that do break through, hopefully we can have these treatments that will eventually lead to as the EHV advisory group says, 0 elephant deaths from EHV.
And that's why we should keep studying the pathology of this disease, because there will likely be, even with a super effective vaccine, there's going to be some breakthrough infections or elephants that haven't had the vaccine, and more treatment options are going to be needed.
Absolutely. It's not one size fits all.
That's right.
All these different options. Trying to see if there's any questions that I have missed. I think we are good. But in general, what can people do to help your work? Do you want to tell a little bit about PEEL Therapeutics or anything like that?
Sure, I can start at PEEL Therapeutics, and then I'll turn it over to Lisa, who again really has been a champion for all of this project in the relationship building, team building, and importantly the science. So PEEL Therapeutics, PEEL is actually the Hebrew word for elephant. We began the company based on this initial discovery with colleagues about the elephant P53, and of course that was 10 years ago, and we're still working, like I just mentioned, how can we translate this elephant P53, and we have different approaches that we're trying. How do we deliver it to human patients, or maybe even other animals with cancer? And what we're doing now with PEEL Therapeutics, with elephants and other colleagues around the country and even the world, is we're establishing these biobanks, right? Because again, it's learning how to use this tissue, how to study it, how to understand it, what's unique, what are the unique properties of animals. Elephants, again, absolutely amazing because of their ability to avoid cancer. But then we also, on the flip side, have had this susceptibility to getting these viral infections and hemorrhagic disease. So what can we learn? And we can only learn that by working together with people on this call and other colleagues. And again, we're really pleased that Dr. Howard is working with us in PEEL. And so, of course, you can contact her as well separately if you want to learn more about PEEL and the biobanking efforts that we're doing. In terms of this research, let me turn it over to Lisa and let her have the last word, because it really has been an honor to work with her and see what she's built with everyone together and with this shared love of elephants.
Yeah, I mean, I'm just going to say that the community has been so supportive, and like the project that we just presented wouldn't have been possible without meeting all these wonderful people at IEF. So thank you guys for bringing us all together. That was so important. In terms of the work we're doing now, to be honest with you, it's challenging to continue it because we don't have elephants in Utah anymore. We do need that fresh blood. So when our paper comes back with more experiments to do, we're gonna probably need some funding to go back to Houston Zoo, or you know, hopefully that doesn't happen and they just love our paper and they'll say, let's publish this, and we won't have to worry about that. But everything that everyone's already been doing, folks reach out to me when they're doing a necropsy. We get those tissues. If we get fresh skin tissues, those are the cells that Josh is mentioning. We can generate that into cells, and so we just isolate the cells from that tissue after the elephant has undergone that necropsy, and then those cells will divide nearly indefinitely in culture as long as we protect them and keep freezing them back. They're not immortalized cells, but if we freeze them early and then we can pull out a new fresh vial of them, we can use them for a very, 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 very long time. And they are teaching us so much. And the cells that we've already generated from tissues people have provided have really taught us a lot about how elephant P53 behaves. We can't use those cells, fibroblasts, for the neutrophil assays because we need heterophils, but they're still giving us a lot of really valuable information about other genes that we're interested in the elephant genes that may be different, not just P53 in elephants compared to other animals.
That's awesome. And I one of my favorite parts about the IEF symposia throughout the years is watching the collaborations and the connections that come out of it. And you guys really are a testament to that. And I know I always love seeing your presentations because it's a new way of thinking for me, and it's just fascinating and wonderful. So thank you so much for all of this. I know everyone really enjoyed your talk, and we're excited to share it on replay because it's recorded. So thank you so much, and thank you for everyone for your insightful questions. I'm going to turn it over to Julie real quick.
Yes, thank you so much. I'd like to invite everybody to turn their cameras on, and let's give our presenters a big round of applause for a wonderful presentation. Thank you so much. It was so interesting and I learned so much. We really appreciate it very much.
And we appreciate you, Julie, and we appreciate IEF.
We do.